Okay, we are ready to start. Welcome everyone, and thanks for joining us today for Neuroelectrics webinar. My name is Rafał Nowak, and I'll be your host today. So this is the 8th of series of Neuroelectrics webinars, and now we have already more than 150 participants. Today we'll be talking about challenges and opportunities of non-invasive brain stimulation in aging brain. So I'm very happy to welcome our guest speaker, Professor Emiliano Santarnecchi. Good morning, Emiliano.
Good morning, Rafa.
Good morning, good to have you here. So just a little housekeeping before we get started. We'll begin with Emiliano presentation with the duration of approximately 45 minutes. And this will be followed by Q&A. So please feel free to submit your questions at any time using the chat tool. And for your information, the recorded version of this webinar will be also available offline. So please let me quickly introduce our speaker, Professor Emiliano Santarnecchi. Emiliano is a clinical research scientist at Beth Israel Deaconess Medical Center and professor of neurology at Harvard Medical School in Boston. His main interests lie in the combination of non-invasive brain stimulation, electrophysiology, and neuroimaging to modulate brain connectivity. He is particularly interested in the application of oscillatory electrical fields like TACS to induce long-lasting changes in brain oscillations. His recent projects include the modulation of gamma oscillatory activity in patients with Alzheimer's disorders and frontotemporal dementia. And he's also interested in microgravity and cosmic radiations during space explorations as a window into neurodegeneration on Earth, so well timed with the Mars 2020 Perseverance mission. So obviously a lot of shared interest with us here at Neuroelectrics. Welcome, Emiliano, and the floor is yours.
Thank you, Raf. I'm going to share the screen. Let's see. There you go. So, okay. Now you should see the screen.
Yes.
Okay, so first of all, thank you for inviting me. This is not the first time I give a presentation for Neuroelectrics. This is probably the third or fourth time. It's always been a pleasure. Today, I'm particularly excited about sharing our adventure in trying to find non-invasive brain stimulation solution to treat or at least slow down Alzheimer's disease and also other related dementias. The presentation is structured in a way that I will give you an introduction about why we do what we do, meaning what are the neuropathophysiological bases of Alzheimer very quickly, why do we adopt brain stimulation as a solution. Then I will show you the summary of 2 quick pilots that we have run over the last 3 years with some preliminary results, and then we will jump into future directions that we want to do next. Obviously, I'm summarizing years of work in 30, 45 minutes, so it's going to be a bit of a rush through the slides, but then we will have the Q&A, and I'm also available to answer any question via email, obviously. 30 seconds about me. As Rafa was saying, I'm an assistant professor here in Boston. I have my own laboratory within one of the major hospitals, PADMC. I'm also affiliated in other departments in Boston, and that's why, as you will see in the next slide, I have this affiliation with other things that apparently don't have anything to do with neurology, but actually like physics. But actually, in nowadays neuroscience, there is a lot of integration between fields, and so we leverage a lot from mathematics, physics, and so on to actually understand the brain as the most complex system we have in our universe. I graduated in this lovely medieval town in Siena, in Tuscany, in Italy, where I was born and raised. I moved to Boston a few years ago, accepted a faculty position, but I'm still very much in touch with Siena, and we try to do a lot of studies and research together. What do we do in my lab? It's called the Network Control Laboratory. We use multimodal approaches to characterize and manipulate brain circuitry. This means that we use everything from neuroimaging techniques, structural MRI, functional MRI, any type of MRI. We also use PET data. We use electrophysiology data, as you can see here. This is an image of a combined TMS, so transcranial magnetic stimulation technique combined with concurrent EEG recording, so we can look at high resolution response of the brain to brain stimulation. And we also use neuronavigation systems similar to the one we use in surgery, neurosurgery. So it's a complex setup. We try to attack the problem from many angles, and you can get a better understanding here. So to do what I call, and I like to think that's what I'm doing, image-guided network neuroscience, we definitely leverage a lot of neuroimaging and connectomics. We look at how the brain is structured, how it functions, how regions talk to each other. But we also use a lot of neurophysiology, so electroencephalography, electromyography, and so on. And then we use non-invasive brain stimulation, obviously, as a way to causally test any inference, any hypothesis we have about the brain. With brain stimulation, you can actually interrupt the circuitry or make a region fire more and gets more activated. And so then you can see behavioral changes related to that change that you induce. And now you know that that region is really important for that cognitive function in a causal way. At the same time, we do a lot of studies on human cognition and cognitive enhancement, also in healthy humans, trying to boost attention, intelligence, memory, and so on. And we leverage a lot of mathematics and complex network theory to understand the complexity of the brain and then decide where to stimulate, for instance, or how to look at the impact of brain stimulation. That being said, at the end of the journey, it's always something that I kind of think I like to do in a way that it's basically falls under translational neuroscience. So we try to use all this knowledge about the human brain and techniques to measure it and modulate it to capture as soon as possible new evidence in animal models and translate them into humans in what we call first-in-human trials. And these can be done in many pathologies. As you will see today, we have done it in Alzheimer's disease, but also in brain tumors, in other diseases when you have a nice new insight, which is a breakthrough insight in an animal model, but you don't know how to translate that in humans because obviously you cannot do the same things in animals and humans. But we kind of specialize in a way in an approach to translate this as fast as possible that can save the translation time from an evidence in animal to an actual, I don't know, drug development in human or a new therapy by a factor of 2 or 3. So instead of waiting 15 years, maybe you get results already in 3 to 5 years. And that's what I'm really pushing for, because I want to get results in humans, a new biomarker discovered to capture disease or to follow the disease over time, but also most important, therapeutic application for patients. But let's start from the problem that we're trying to address today, which is Alzheimer's disease. As you can see here from the numbers, it's what we can call a silent epidemic. Basically, it's the 6th leading cause of death in the United States, has an estimated cost of $300 billion in 2020. In 2050 will be $1.1 trillion of cost. If you think about the different causes of death since 2000, meaning 20 years, a 20 years window, there has been a decrease of 7.8% of death caused by heart disease. But in terms of Alzheimer's disease, there has been an increase by 146% of death caused by Alzheimer's disease-related dementias. It's a huge burden to society, to the patient, obviously, to the caregivers. It changed lives, it changed families. We definitely need to find a cure. But the most disappointing thing is in this report from the Alzheimer's Association that recognize that none of the pharmacological treatment that have been tested today, even though we've spent billions and billions and 3 decades on this, none of them are actually really effective in slowing down the disease on a mechanistic level, and so being able to change the disease course. A lot of them just boost cognition, maybe, but really we are missing something, and we are losing hope in the search for new therapeutics for Alzheimer's disease, at least based on pharmacological therapies. The problem for me is that we always look at this problem from the typical type of information that we used to have in the past 20 years or 30 years, which is mostly neuroimaging, MRI or PET. And so we know what's, let's say, wrong in Alzheimer's disease. This is a nice paper from 2011 summarizing what are the typical alterations in Alzheimer's disease. You have alteration in functional network in specific region of the brain. You have accumulation of proteins that tends to co-localize with alteration in networks. So all these things tend to happen more or less in the same region, but they all come from different neuropathological substrate. You have also atrophy, so the gray matter in the brain gets thinner. You have hypometabolism, so this region receives less glucose, and so meaning they are less active, and the metabolism goes down, and so definitely the functions that are supported by this region tends to decrease. You have alteration in the memory network. Obviously, that's one of the main symptoms of Alzheimer's disease. But everything has always been defined, as you can see, from a brain structural or functional perspective, so using neuroimaging. But what was missing, and finally, over the past 6-7 years, we've been seeing more and more publication on this, is the mechanistic level of alteration in Alzheimer's disease, the neuropathophysiology at the level also of electrophysiology of Alzheimer's disease. And so look exactly at which neurons, which cells in the brain are hypoactive or hyperactive, what kind of oscillatory activity they generate, and an electrical level, not just an imaging level, neuroimaging level. So big pictures of the brain and big areas of activation. Here we can look finally at which subpopulation of neurons actually have maybe a deficit, and that's why Alzheimer patients show the deficit that they show. So long story short, over the past few years, this is a fantastic paper by Jorge Palop summarizing network dysfunction in Alzheimer's disease. We have come to know that there is a specific class of inhibitory interneurons in the brain called parvalbumin positive interneurons. These are not excitatory neurons. These neurons are in charge of inhibiting the activity of other neurons, which seems a counterintuitive thing to be responsible for Alzheimer's disease. But we have found that these parvalbumin positive interneurons actually are less active, and they go through neurodegeneration, obviously, and cell death. And when these neurons are actually active in an healthy brain, they generate a lot of activity at 40 Hz. So there is a specific rhythm that they fire at. And so they generate this fast oscillatory activity. It looks something like this. And we know that in Alzheimer's disease, also in patients, there is a deficit of this activity. So finally, we have an idea what could be the oscillatory activity that we need to restore, and what is the class of neurons that we need to restore. We know that gamma oscillation, this 40 Hz oscillation, falls into the so-called gamma band activity measured with EEG, also in humans. We know that this activity has some sort of a protective value against epileptiform activity. So if you have high gamma, you have a less likelihood of developing seizures, which are very common in patients with Alzheimer's disease. And so that makes sense in the sense that you have less gamma, you have more epileptiform activity. And gamma is also supportive of a lot of high-order cognitive functions like memory, attention, abstract reasoning. So this could also be partially responsible for this cognitive deficit. So there is a new idea now. We need to go after these gamma alterations. And this paper was from 2016. So it's relatively recent. But what was really impressive that year is that another group from MIT published a very influential paper showing that actually in an animal model of Alzheimer, if you try to restore gamma activity exactly at 40 Hz by having these animals be exposed to a flickering light that flickers at 40 Hz, so a light on the screen, black and white, that change very fast, like 40 times per second, you actually see a spike in the activity at 40 Hz in their brain. And most importantly, when you actually look at, for instance, the protein in the brain, like amyloid beta, what we consider the main problem of Alzheimer's disease, at least until a few years ago. Now we think it's more tau protein. But in any case, they show that there is a decrease in amyloid beta when the animals receive 40 Hz stimulation. And that was not present when they were in a control condition or they receive stimulation at random frequencies. At the same time, they were also able to show that this is not just a random effect. Before you see that, you see a lot of activation of microglia. And this microglia is what is in charge of neuroinflammation in the brain, and meaning it's an alarm sign when you get microglia activated, because microglia now knows that there is something wrong in the brain and it's going to go and try to fix it. And in this case, they were able to show that actually this microglia cell migrate towards amyloid plaques in the brain, and they start metabolizing them, eating them, basically. That's the role of microglia. And that's why you see this decrease in amyloid beta. So this was a fantastic finding in animals published in Nature. The following year, they showed that there is also a decrease in tau protein, which we think is the real responsible protein for cognitive deficits in Alzheimer's disease. They also show some memory improvement in these animal models. So this seems to be like the way to go to finally find an alternative therapeutic for Alzheimer's disease. So restoring gamma oscillation could be a new therapy. Obviously, the problem there, as I mentioned before, is how do I move from this to this? They did stimulation with flickering lights, but they also did a very invasive stimulation, which was probably the most effective one, by injecting viruses in the brain of the animals with invasive surgery. So it's not always easy, almost never easy to translate the efficacy of an animal model into humans when you cannot do all those invasive procedures. So that's where brain stimulation came into play, at least for me. At the time I saw the article and I was showing a few slides, I was already working on inducing gamma oscillations at 40 Hz in healthy controls, just to boost memory, attention, intelligence, and many other cognitive functions. And so I kind of already had an idea of how to do it. And for those of you who are not familiar with brain stimulation, we think that there are two big subfamilies. One is composed by transcranial magnetic stimulation techniques, which are considered neurostimulation technique. As you can see there, the devices compared to the one we use for electrical stimulation on the right are bigger, bulkier, more expensive in general. You do these techniques mostly using neuronavigation, so you need patient MRI. It's a complicated setup, but at the end of the day, it's very effective, and it's TMS is FDA approved for the treatment of depression, migraine, obsessive compulsive disorder. So it really works. But to induce high frequency activity, this is not the best technique. And we tend to think that the best solution would be to use a neuromodulation technique, neuromodulatory technique, like transcranial electrical stimulation, where the stimulation is not as high intensity as TMS, but you can actually play, if you want, with frequency. And just to be more specific, there are different types of electrical stimulation, but for those of you who are familiar with this, you will recognize here on the right, transcranial alternating current stimulation, which basically is one of the three main component of the TES family, transcranial electrical stimulation family. But what TACS brings to the table that the other technique do not is that you can actually add a frequency component. So now you can induce an electrical stimulation in the brain by electrodes placed on the scalp. As you can see here, there are many devices that allow you to do this. Devices with joint 2 sponges, devices with multiple electrodes, and so on. And when you add the frequency component, you can actually now think of engaging neurons that tends to oscillate at that frequency, let's say 40 Hz, and boost their activity. So this is not new. We have been playing with this for 20 years. I keep moving my window because I'm not sure you can see the screen. This is work from a good friend of mine, Flavio Frolich, from 10 years ago, when he showed that when you look at freely behaving neurons, in this case, this black line, you have a neuron that's in a random way sometimes start firing and increasing activity in response to stimuli or anything else. But when you overimpose this oscillatory activity on top of the activity of the neurons, you actually get activity that now start matching the peak of the oscillatory activity that you induce from the outside. When you increase the frequency of your oscillation, you also get faster activity in the neuron. You increase the oscillatory activity even more, and you see that the neurons tends to follow. So if this frequency was 40 Hz, you would have induced the neuron firing at 40 Hz, which is what we want. As I mentioned before, I've been playing with this idea of modulating activity in the human brain and the gamma band for many years, starting with my PhD. So I had an idea of how to do it, and I was confident that this could work. The problem was, okay, if you do it in Alzheimer patient, to translate this finding right away, how do we do that? So where do we stimulate? Now we know the frequency, but where do we stimulate? So this is the same image I showed before. There are many options to stimulate in an Alzheimer brain. But obviously the problem, we think the main problem is protein accumulation. So amyloid and also tau are the main proteins that we think we need to help the brain get rid of. And so we decided to go after amyloid to begin with, because that was the first finding from the animal model. To do that, in my lab, we come up with an ad hoc PET analysis pipeline. PET, for those of you who are not familiar, is a neuroimaging technique, actually falls under nuclear medicine, but it's another imaging technique to look at the position of proteins in the brain, but you can also look at metabolism and other aspect of brain function. You do it with an injection of a radioactive tracer, and then you scan the brain and see where the tracer actually binds more with neurons and tissue. And that reflects the fact that in those locations, there are more amyloid beta if you use a tracer that is sensitive to amyloid beta or tau, or there is a decrease in metabolism and so on. So we came up with a way to extract more information for this PET data. It resulted in these very nice high resolution maps of, in this case, amyloid beta deposition. This is a template patient from my sample. You can see that amyloid tends to accumulate more in the temporal lobe. You can see the red values in the south side of the brain and also in the frontal lobe. So now we know more or less where we should stimulate. You can also see it here exposed on the white matter. It's easier to see that it's frontal and temporal and so on. So now we have a map. You can see it even better here. Now we can threshold this map and locate the region when you have more amyloid compared to the rest of the brain. And in this specific patient, you can see that we remain with something in the temporal lobe, on the right hemisphere, and on the frontal lobe. So now you have two target maps, and we can actually now personalize the treatment using multiple electrodes placed on the scalp so that we induce more 40 Hz modulation in the region when this patient in particular has more amyloid, and we try to spare the other region of the brain. These are 3 example patients when you can see that the position of the electrodes are all different because they all have different maps of accumulation of amyloid or tau. And so you end up with this first attempt of personalized brain stimulation for Alzheimer's disease based on these amyloid maps. The idea, more in general, when I launched the first trial, and you can see here, we were really, really lucky to secure money. I was able to win an award at our hospital at the end of 2016, beginning of 2017, only a few months after they published the paper in the animal model. And with that money, we actually started the first trial. I got approved to test this 40 Hz modulation in 10 Alzheimer patients with mild to moderate Alzheimer's disease. So it's an advanced stage. But the main idea has always been to replicate everything we saw in those models from the first paper when they discovered this gamma alteration and inhibitory interneurons to the animal model when they show an increase in microglia. So we think we have kind of all the elements of the pathophysiological cascade of Alzheimer's disease that includes alteration in inhibitory interneurons, alteration of gamma rhythm, neuroinflammation, change in amyloid beta, change in p-tau protein. And the idea was, how can I translate this in humans? How can I measure the same thing? So our study design gets a bit crazy in a sense because I wanted to really cover all the angles. And so in a subsample of the patient that we did, we end up doing 3 types of PET scanning before and after the intervention with TACS. We did amyloid beta PET, tau PET, and also microglia PET using experimental tracer that measure microglia activation and neuroinflammation in the brain. We did MRI, any type of MRI, structural, functional, cognitive testing, resting state EEG, blood markers, TMS-EEG, many other assessment. And we then expose this patient to either 10 sessions or 20 sessions of 1 hour of TACS every day in a span of 2 or 4 weeks. So at the beginning, we didn't want to go for a longer treatment, but these are already doses that are way higher than anything else that has been published. Usually you do TACS for 20 minutes, 40 minutes, maybe once or 2-3 times. In this case, we are talking about 10 hours in total, or 20 hours. So it was an intense treatment. Patient came in every day at the hospital. We pay for transportation. We try to keep it as simple as possible for them. And we did everything, all this stimulation session, and then we repeated the assessment with, again, the 3 PET scanning at the afterwards and MRI and so on. This is a summary of 2 trials. Some things were done in 1 trial. For instance, another trial didn't have microglia PET, but this is just to give an idea of how we approached the problem at the time. The idea of having also microglia, you can see here, we team up with MGH, the Gordon Center for Biomedical Imaging, where we could do amyloid beta PET, tau PET, but also microglia PET. And I'm going to show an example of a few patients, like 4 patients here, when you can see how from different patients you get different deposition of tau protein here. And you can see that tau tends to spread more in the temporal lobe, but like this, but certain patients also have a more diffuse accumulation of tau. Now you have amyloid. All the patients were supposed to be amyloid positive to enroll in our trial. So we did PET, and you can see they have a lot of amyloid. Obviously, that's to be expected in a mild to moderate Alzheimer's patient, but also microglia. This was the first time, I think, that on the same patient, you have the 3 tracers. And you can see that microglia show some inflammation. You should always look at red and yellow, mostly red. But in some patients, it's more in subcortical structure and nothing in the cortex. Some patients show some inflammation in the temporal lobe. Again, some patients show a bit more, but it's not very diffuse in the brain. So this was fantastic for us to see, because now we have a measure and we can see if neuroinflammation really changed when we do TACS at 40 Hz. That's why I want to go into the future direction section very quickly. The first message is that even though we are at the beginning for me, but we speed up the process a lot, we have learned something. And I think the community is realizing this, is realizing the fact that non-pharmacological intervention for Alzheimer's disease are getting basically traction. They're valuable, they're promising, and we shouldn't just look at drugs for Alzheimer's disease. These are two interviews that I gave for Nature in 2018 and PNAS earlier last year, when we discuss together with other people that are doing the same thing, including the group at MIT, what is the future of this technology approach for Alzheimer's disease. And there is also this nice aging and brain health seminar when I'm invited. There is also the senior author of those amazing papers from MIT, Dr. Li-Wei Tsai, someone from Johns Hopkins and Oxford University, to discuss about neuromodulatory intervention in cognitive decline. So finally, we are creating kind of a movement, and we think that this approach is actually very valuable because you can do more personalization, you can do many more things compared to drugs. Obviously, a combination of the two would be also ideal. So I invite you to attend this seminar. Where are we going in terms of future direction? We have now a bigger trial that is going to end in 2023 from the NIH here. In the US, when we repeat a lot of the things I showed you before, amyloid and tau PET before, we also do different doses of TACS. We expose people to 2 weeks, 4 weeks, 4 weeks twice a day, and so on. We have a sham arm, obviously, a follow-up after a few weeks from the end of the treatment. At the same time, as I mentioned, we are exploring sleep. So we know the protein clearance takes place mostly during the night, in specific sleep stages. So we published these 2 reviews last year, try to understand how to do it and what's out there that people have already tried, not in Alzheimer's disease, but in general, to use TACS to modulate sleep waves. And so that's definitely something to be explored. We are also pushing for what we call non-invasive brain stimulation 2.0 or 3.0. We have this paper now in the review with this big selection of experts in the field that I was able to bring together to basically move from what we know that has been done in Alzheimer's disease so far with TMS or TES. And as you can see, people have stimulated very specific region. There is no network approach, no protein targeting, and so on. And we try to move it towards the next generation of brain stimulation, where we use a lot of biophysical modeling, neuroimaging data, electrophysiology to guide stimulation. This is just an example. This is part of a grant that we have. Neuroelectrics is part of it, obviously. They are the leader in the team. Many other partners, 6 or 7 from Europe and my lab here in Boston, when we try to basically create a virtual Alzheimer brain, meaning we try to characterize the brain of Alzheimer patients from a modeling perspective, look at single neuron activity, assembly of neurons, specific region in the brain, and then up to the entire brain and functional networks. This way, we think we can actually simulate brain stimulation and see how different patients would respond. It's a long journey. The project just started. It's going to take 5 years, but that's definitely the direction we want to take to bring new tools to non-invasive brain stimulation. I think this is one of the last things, and then I'm done. What I've realized over the past few years is that the more I look at this gamma alteration and the specific activity of interneurons in Alzheimer's disease, the more I find traces of the same thing in many other conditions, like autism, schizophrenia, other type of dementia. So definitely there is something there. This therapy could be applied to other conditions. And indeed, we just started a trial here in Boston with collaborators at MGH and also a team in Italy, in Rome, to do the same 40 Hz intervention for 6 weeks in patients with frontotemporal dementia. And the idea there is the same. We know from the literature there is an alteration of a specific class of inhibitor interneurons, and so we can go after these somatostatin interneurons and try to induce an effect similar to what I showed you before for Alzheimer's disease. Quick diversion. We also do this, for instance, in brain tumor patients. This is another area when we have used a lot of imaging and clinical decision on how to use imaging to optimize the stimulation template to modulate, in this case, perfusion, the same perfusion I showed before, but inside brain tumors, glioblastoma tumors. And we were able to show, this is from summer 2019, that we were able to optimize this, and in patients with brain tumors before they went to surgery, we were able to do stimulation inside the MRI scanner and show a decrease in perfusion in the tumor, which might lead that there are less nutrient going into the tumor, and meaning that you can slow down metabolic activity and proliferation of the tumor. Obviously, everything I show you is not just me. I have the crazy ideas, and then we need a lot of smart people to put them in action. So I have a big team. Some of the people here are not part of my team anymore, but they were related to some of this work. There are new people that are not here, but it's definitely a team effort. And these are some of the funding agencies that support our work. Last thing, we have some job opportunities. I have 2 postdoctoral fellowship positions open and some research assistant position open to work on this project and something related to this project. So if you have, if you're looking for a job or you know people, feel free to send me an email and we can discuss. Thank you. I think this is the end.
Thank you very much, Emiliano. Thank you for a great presentation. Thanks for promising results and actually for trying to translate these results from transgenic mouse, from the animal model to the human applications. So we have time for to take some questions. Meanwhile, we are waiting for the question from the audience. Let me just ask actually the main question. So my question would be, yeah, why 40? Why 40 is so magic? So we know that there are different oscillations. We have different models of gamma activity generation, inhibitory neurons, inhibitory excitatory neurons, and we have evidence of 40 hertz in mouse models, but doesn't mean that 40 hertz will work for humans as well. And we have this sensory stimulation, but this again, right now in mice model. So could be that we need different frequencies for humans.
Oh yeah, absolutely. I mean, that's one of the biggest care I have, like the biggest worries. As happened with many of these techniques, like you start with something, you translate something from animal models, or you come up with an idea, you start exploring the parameter space, but then you find out that just to test one parameter, it takes 5 years. And so what you end up with at the end of the journey is definitely something that is not optimal, meaning you should have done more testing than testing other frequencies, maybe personalized gamma frequency for each patient. You can see that they have a deficit in a specific frequency, test the target stimulation frequency. But sometimes you run out of time to test all these things because there is a need to for approval of a therapy. So now I'm trying to think of ways to actually explore other frequencies, but as you can see, the results I'm reporting now are from ideas from 2017, and it's a long journey. For me, it was obvious at the beginning. We need to personalize. Maybe it's not just gamma. Maybe it's not just 40 hertz. Maybe you need to do stimulation in theta, because theta and gamma are related. And so maybe just stimulating
cross frequency
gamma. I don't have an answer, but I'm pretty sure that you're right.
Okay, and actually the second question is because you were mentioning that you are trying to stimulate during sleep. As far as I remember, that this CCSF clearance during sleep is somehow related to non-REM sleep and slow wave activity. So how you relate this? There is a correlation between slow wave activity and level of clearance during sleep. And you will apply gamma during sleep as well, or this will be the slow waves?
We are testing slow waves. Okay. So again, it's another example of another answer to your question. Like it's not gamma everywhere because we like gamma or 40 hertz. In that case, it's low wave sleep. We try to put people asleep faster, so to induce sleep, or to boost sleep stages. And in that case, you use low frequencies. There is also some evidence for gamma in deep sleep, but it's mostly slow waves.
Yeah, and especially that there is like mystery where the clearance occurs during sleep and not during wakefulness. So why during wakefulness we are not clearing, we are not throwing our waste from the brain. So let me quickly.
I think there are a few Q&As.
From the first one from Michael, actually asking of the bands for gamma you are using during this preliminary results analysis. So it's the question of the definition of gamma bands, and especially regarding the high gamma.
So we do what I decided to call narrow gamma is basically 38 to 42 hertz, just around the 40 hertz. I want to see if there is an effect there. Then you have some low gamma, which is the boundary of theta, of beta. So let's say 35 to 50. And then you have, I think we did 50 to 70 and 70 to 90. Okay, I can check if they send me an email, I can give the exact number.
And especially that actually this high
we don't see much in the super high.
Yeah, but I mean that this is actually the most difficult to register actually with EEG. So I was also surprised that you were getting so clear evidence.
We spend a lot of time cleaning this data and reviewing the data. We do a lot of artifact templates at the beginning, like we ask patients to clench their jaw to measure some muscle activation, and then we use templates to remove it. I mean, I know it's definitely noisy data. That's another problem with gamma.
Okay. Okay, so let's go to the next one. Here is only regarding the traces, so let's skip this one. TACS intensity, if you can just provide some parameters for the stimulation. Like 40 hertz, we know already.
No, we use for 2 milliampere per electrodes as a maximum. And when we do this multi-electrode solution, is obviously 4 milliampere total. But in some patients, when we stimulate the temporal lobe, they cannot tolerate 2 milliampere. And so we end up going about 1.4, 1.5. That's kind of the magic number that they can tolerate because it's very painful on the temporal lobe. There is a lot of muscle here.
And especially for TACS.
Exactly.
Comparing
so we tend to adapt it, let's put it this way, and you have to do it with these patients.
Okay, question from Santiago. Related to your results involving microglia and protein accumulation, you see a reduction in the protein accumulation and microglia. Could this mean that there are other mechanisms leading the protein degradation activated by the gamma stimulation? So it's like the mechanism of gamma clearance, if it's epiphenomena or it's like the mechanistically.
I mean, the only thing that makes me think that is not an epiphenomenon is that the data I showed were extracted exactly from those yellow areas that you saw in the MRI, which is the modeled induced electric field from the montage we used. So we only extracted from the region where we thought this should happen, the effect should take place, and we saw the effect there. When we use other region like the motor cortex, when we didn't stimulate, or visual, you don't see that change. So I think it's something that is happening where we stimulated, so probably it's related, but I'm gonna be very honest, we don't know. Especially why microglia goes down instead of up. I mean, it could be, I don't know. I'm gonna be very honest here.
Okay. Question from Alberto. If I got it right, with TS you can both increase and decrease perfusion. So it's with regards to the part you were talking about changes in perfusion. How does it work? Do you use different stimulation protocols to obtain this opposite result?
I didn't get the first piece. With TSCS, you can both increase or decrease?
Yes, so it's the question actually.
Oh, okay. So as far as we know, the reason why we were looking at perfusion is that there is a nice paper in Neuron from 3 years ago from Matteo and colleagues, when they showed that actually 40 Hz oscillation measured with invasive electrophysiology correlates positively with the diameter of vessel in microvessels. So the more perfusion, the more firing in the gamma range. And they were actually able to show, and it's a fantastic paper, that there is a relationship there. So if you increase gamma, you should expect to see more blood flow. That's what we are measuring in the paper as the substrate. If you could get paradoxical effect, I don't know, like also a decrease, it's tough to say. But based on the model that we use from this paper, that now has been replicated and so on, that was the expected effect.
Okay, the next question from Simone, actually he knows you very well. Do we know how many astronauts have developed Alzheimer's disease?
That's a good question. I don't think we will ever know. We will never get access to that data. No, there are reports actually. There are reports from one of the twin study. One twin, there are twins astronauts.
Oh, yeah.
One went for 1 year to the International Space Station, one stayed on the planet and on this planet. And they measure cognition, and there were some differences, also changes in mood, obviously. It's difficult to get access to that data. But I don't think anybody developed full-blown Alzheimer. But the goal here, why I mentioned Mars, is that all this evidence is for 3-6 months in space. Now, to go to Mars, it's going to take 1 year to go. 1 year you stay there, 1 year you come back. So it's going to be a 3 years exposition. So in that case, I don't know what could happen. So again, let's not make it a big deal and alarm everybody about this, but it's something to consider.
Yeah, no, obviously, we're happy that there is someone studying this already. Next question from Michał. It's regarding actually how to combine, or we actually should be combined the sensory stimulation, like flickering or auditory stimulation, the 40 Hertz, with gamma TES. So combining the sensory stimulation with different modalities and transcranial electrical stimulation.
Yeah, just something I didn't mention, but we are also running a small pilot with flickering lights and auditory stimulation. So it's not that I believe in TSES and I don't believe in the other approach. I think it has a lot of value. The reason why I'm not pushing completely for it, or let's say the reason why I think TSES as an edge is because you can target it everywhere. With visual and auditory stimulation, you are likely only engaged in sensory region. I know that in the animal model, they show that it spreads everywhere, but the brain of a mouse is way less complicated and clarified, and so things tend to spread a lot. In the human brain, our sensory region are very confined, and so I don't know if it's gonna work the same way in humans. Combining them, if you can keep track of the phase, and so you don't create actually out of phase stimulation. So when TSES is at the peak, your sensory stimulation is at the trough or something like that. If you can manage that, probably there is an advantage.
You will get a double effect. But actually, so the goal, for example, in frontotemporal dementia, or if you will use for primary progressive aphasia with the clear pathological lesion or area, at least for stimulation, you will use like more focal stimulation, even with TACS. But how to translate this to, for example, to Alzheimer's, where the accumulation of beta amyloid is spread over the brain? So do you assume that, like with sensory modulation, there will be somehow
that's the results and the predicates of the animal model. They stimulate with flickering lights, but then saw an increase in gamma at the beginning in the visual cortex and then everywhere. But again, that's a brain that tends to be more connected from an evolutionary standpoint. Yeah. In our brain, if you look at the visual cortex, we have a giant sulci between the visual cortex and the rest of the brain, and that's for a reason. So activity in gamma locally in the visual cortex doesn't necessarily spread very easily to the surrounding cortices.
Yeah. So could be that the answer would be actually multifocal stimulation with different regions simultaneously with gamma activity. Next question from Theodore. What were participants doing while TACS was applied, and actually how do you control the drugs in Alzheimer patients? Next question from Daniel.
That's a good question. So that's actually something I didn't mention, but it's a crucial factor here. As we know, TACS seems to, like any other type of brain stimulation, to rely a lot on some brain state dependency of the effect that you see. So you cannot just stimulate whenever you want and pretend that everything is going to work. Especially in the case of oscillatory activation, stimulation, you want to be sure that the brain is in a state when it's either already producing those oscillations or is in a state when it can kind of accept this entrainment. So it's difficult to predict what the state could be. What we're trying to do is to keep everybody in the same state. But since this is a delicate population, Alzheimer's disease patients, they stay with us for 2 hours every day or 3 hours. We don't want to ask them to fix a crosshair on the screen. I mean, that would be impossible. They will also fall asleep. So we have them watching documentaries. We have a predefined set of documentaries. We offer them a choice every day, and they watch something together with our research assistant. So it's engaging. They are in the same state, but maybe they're not thinking about anything else. That's as much as we control brain state. I always like to go for feasible things that I could do in an actual patient tomorrow. Maybe the patient will receive stimulation on the sofa when they're watching TV. You cannot ask them to do 2 hours of watching at the dot on the screen. They're not going to do it. So
but you assume
that's what we're doing.
So that one day we could apply this at home.
In that case, you can come up with an app that have cognitive tasks that they do all the time, which is something we are developing. When you can fix the state by doing something actually meaningful for them, they might actually leverage gamma. You do a memory task. or you can have them do whatever they want. I mean, we need data to say if it's really important, this brain state argument or not. To me, it is. So I try to find a compromise.
Okay. We have some questions from Jason, actually regarding the benefits of TACS over light, sound, and other forms of stimulations. Actually, you have answered this right now. The neurofeedback training, if you can get some similar effect comparing to TACS. And here the question actually from Elise, who's surprised in the animal model, did 40 Hz stimulation lead to the microglia clearing up to the amyloid in the animal's brain? So actually she's referring to the seminar work from Tsai from MIT. So yeah, so yes.
They didn't measure during the measure afterwards. They had to sacrifice the animal, obviously, but yes. There was microglia activation, amyloid clearance, and then also tau.
There is a request from Vanya. I'm a clinical neurophysiologist for stimulation parameter based precisely in what exactly stimulation intensity train, intertrain time, etc. I think here we can refer to all your papers, and there you can find all information regarding the technical details of the stimulation. And we have to mention that this is on the research level still, and
yeah, I mean that's and it's super important message to mention. Yeah. These are all preliminary results in a research environment with selected patients. Like let's not jump to conclusion that anything is working here. I just want to make it very clear. Like this is a very interesting, promising avenue, but we are at 10% of what we need to do, I think.
So we have more questions like this. With TACS, where is the right location to be effective, and what are the right locations? So
if I knew, I wouldn't tell you. I would do it myself.
Yeah, but
no, I'm joking.
Is that obtained from PET imaging? So actually, so
yeah, I mean that's why we do all this. Yeah, that's why we do all this imaging, to try to get an informed decision based on some data instead of just putting electrodes on the scope. But the right answer we don't have. We don't know what is the best location.
Actually, a little bit technical question is, for the protocols in the Alzheimer, it is enough device with 8 electrodes, or do we need 24 electrodes? So it will probably depend.
What we use now is a maximum of, I think, at the beginning it was 8 or 10 electrodes, but then I decided to reduce it to very few electrodes, because also technical difficulties during the session with these patients, like they move a lot. So we didn't want to have to spend a lot of time refixing the electrodes and so on, for impedance problem and so on. So sometimes we use 4. It depends on how many regions you want to stimulate. So definitely you don't need more than 8 or 10. The 24, we use actually 32, but we use them for EEG recording before and after.
Yeah, okay. So it's if you want to combine EEG and stimulation, you should use more channels, obviously. Question from Felipe. Are you using any kind of computational neuroscience approach to better explain the mechanism that you want to target? So target actually.
Yeah, that's that project that I mentioned. We're trying to build this virtual brain, not just to predict the response to stimulation, but also to define personalized stimulation parameters and so on. But we are far away from having a model, to be honest. So we don't have a tool yet. We are building it, and I like that approach a lot. So definitely we need to have some computational tool to say, okay, if I actually change this channel, or if not channel, I mean calcium channels or something like that, or if I shut down GABAergic interneurons, what happened? Does it look like the effect I'm seeing or not? So you can retrofit your data and then get an hypothesis or an explanation. But for now, we don't have that.
Okay, next question from Roger. Tau seems more important than amyloid beta. Moreover, some amyloid beta do not have parvalbumin positive for PET detection. Why not designing the targets based on tau deposition?
That's what we're doing now in the trials. So we also get tau before, and that's why I mentioned we end up using less stimulation electrodes. And as you can see from the daily visit, we also have subjects that receive stimulation basically just on the temporal lobe, bilaterally, because that's the decision I made a couple of years ago, maybe a year and a half ago, to maximize stimulation there, because you also have amyloid, but you also have from the microglia PET, it seems the inflammation is mostly there, even though it's very experimental, very experimental tracer, but also we know that tau deposition is this plus other regions.
Okay.
So tau is the only thing that correlates with cognitive deficit. So definitely that's the target.
Okay, question from John. Actually, if you could not personalize stimulation targets with PET, what would be your choice locations to target TACs in MCI and Alzheimer's disease?
If you cannot personalize, I mean, you can still base your decision on published data, which is what we did at the beginning. I mean, then I got access to the data by scanning the patients, but at the point I already got the IRB approved and the grant approved. So my guess was I look at an average map of where tau is in the Alzheimer's brain, and then I put the electrodes there. We were also able to do personalization, but it's very expensive, as you can imagine, to do all this PET imaging. So if you cannot do it just based on available data, there are studies with hundreds of patients.
Or actually choose a group that doesn't have to be Alzheimer's, but for example, like behavioral variant of frontotemporal dementia, that it's quite easier to target and to find the target than spread.
If you look at the definition, diagnosis, and everything, you should still have some sort of PET data, an FTG PET to look at hypometabolism. If it's only frontal, only temporal, so you always end up, if you want to do things like 100% accurate, you always need some imaging, including PET. But again, we know what is the average map from a behavioral variant frontotemporal dementia patient. Your patient specifically that come in tomorrow might be slightly different, but yeah. Again, we need to start from something.
Okay, a question from Caroline. Did you observe any behavioral effects on these patients?
That's actually a good question. So behavioral, I mean, both cognitive and actual behavioral effects. So something I didn't mention, we give them an actigraph, an ActiWatch, to look at sleep patterns and how they behave during the day. They wear it from the very first day until the follow-up, so like for 2 months. We are analyzing the data now. I don't have an answer, but definitely something to consider. From cognitive assessment, we didn't see striking changes, to be honest. And also, I think that was part of the design. At the beginning, it was more of a safety and feasibility study. You cannot expect to see huge changes in cognition in 4 weeks in an Alzheimer's patient. If you see them, there's something wrong, or something to be investigated more comprehensively. But yeah, we don't see, nothing goes down, so there's no detrimental effect, but there's also no huge change in ADAS-Cog or MMSE or, yeah. Okay. That's why we need to do 3, 6, 12 month studies.
Okay, next question from Rosé. It's actually related to the function of microglia, microglia activation with gamma, but not with alpha oscillation. If you can say something about this, that actually it doesn't work with alpha oscillations, but with gamma.
I don't know. We haven't tried alpha, so I don't know if she's referring to a paper. I don't know.
Okay, so we can check this, but yeah. So it's like the question from the beginning: why 40? Why 40? So we assume that the 40 should somehow shape the spiking on the neuronal level and then generate gamma. But
if you go after microglia, it could be many other things. I mean, we know that there is a relationship between inhibitory interneurons and the fact that they trigger microglia activation when they work properly. So that's kind of the chain of event we are trying to inject. To evoke, but again, it might be that there is a more direct way to activate microglia, and that's it. You don't need to go after inhibitory interneurons.
Okay, and
there is a lot of things that we don't know. So again, I have to say it again, otherwise people might get like an idea for the design tomorrow morning, and they think, oh yeah, based on the Emiliano's data, this is gonna work.
So here comes another question from Elise. If this could be used actually for other neurodegenerative disorders affecting more subcortical areas, like Parkinson, for example.
We are not looking at Parkinson. We have a few discussions internally, but we're already busy with Alzheimer and frontotemporal dementia. Maybe. I mean, if you have a strong idea at this cellular level, again, some evidence of which class of neuron and interneurons might be affected. If you see, again, it doesn't have to fit with this idea, with the story of 40 Hertz and this emitter into neurons. I mean, it could be that in Parkinson's you need to do something else. So.
Yeah, actually, she proposed here cortical subcortical pathways to stimulate and to have as a target.
But there are no strong data with electrical stimulation that by stimulating cortex you actually
that you are getting into.
There is more evidence with TMS, which might be the therapy in that case. You stimulate the angular gyrus, you get the hippocampus, that's shown in a science paper a few years ago. You can do it from motor cortex or
trying to go deeper through the network.
Yeah, I mean, again, if you have any question, I see there are a lot of questions, but I love to talk about this stuff. You can also organize Zoom calls, send me emails. We can strategize about your studies. I love to see this expanding in many directions because I'm not able to cover all of them, and I might have ideas that are valuable.
Be careful, Emiliano, because you will have your email box.
No, I love it. I love it. I mean it. Otherwise, I wouldn't say it.
Okay, so let me ask the few last questions regarding from FORI. If you notice any difference between patients who have 4 weeks of TACS versus 2 weeks, and there is a difference regarding the duration.
In the analysis of perfusion, yes, we see maybe a stronger effect for the 4 weeks. I mean, it's double the dose of the 2 weeks, so it's not surprising. We also did a dose response effect, meaning some patients miss some visits. Actually, in that group, 9 visits out of 200. So that is also speaks to in the direction of high adherence to treatment and feasibility of the treatment. But some miss a few visits, so we account for that and we calculated total injected amount of current for each participant. We corrected for that, and regardless of all these corrections, the one in the 4 weeks group tends to respond more, especially for the bitemporal stimulation. So I think, again, not an absolute rule. It might be that in some patients you don't need to stimulate longer. You actually need to do an interval thing, like 3 days stimulation, nothing for a week, another 3 days. We will never know until we test these things, but don't assume that more is better necessarily. As you know, from brain stimulation, there is always an optimal spot. In terms of intensity, it's not always good to go to the higher intensity, but also in terms of exposure and dose, that might not be the case. So test different hypotheses yourself and different durations.
Okay. Thank you very much. So actually, this was our last question. Thanks, Emiliano, for the presentation. Thanks for all answers and talks. Thanks for all questions. I also want to thank everybody for joining us today. So as I've mentioned at the beginning, the webinar has been recorded and will be provided by email to all participants. And I hope you will join us again for the next webinars. You will find the information on our webpage, neuroelectrics.com. So there you can find information about upcoming webinars. So once again, thank you very much. Have a great day and take care and goodbye. Thank you, Emiliano.
Thank you, Rafa. Thank you, everybody in the audience.